In today's lecture at Medical Sciences, we want to discuss together about macrocytic megaloblastic anemia. In the introduction of anemia under classification, we saw that the macrocytic pneumochromic type of anemias, they are categorized into two, that is megaloblastic and non-megaloblastic. So in this session, we are going to zero on megaloblastic causes of macrocytic pneumochromic type of anemia. So we're going to see what is the definition of macrocytic megaloblastic anemia. Then we are going to see what causes it. Those are the deficiencies, whereby it is majorly vitamin B12 and folate deficiency, whereby we're going to look at absorption and metabolism of these folate and vitamin B12. Then we shall look at the causes, signs and symptoms, and we shall explain much on the laboratory diagnosis, how you can confirm megaloblastic anemia. So don't miss lab diagnosis, it is very key, and remember to subscribe if you haven't. So megaloblastic anemia, which is a type of macrocytic pneumochromic type of anemia, is characterized by impaired. So this type of anemia is characterized by impaired red cell maturation, or what we can say impaired DNA synthesis. And when there is impaired DNA synthesis, the nuclear maturation during erythropoiesis, nuclear maturation is impaired. There is no nuclear maturation. So we are going to see, due to impaired DNA synthesis leading to decreased cell division or nuclear division, we are going to see the cells are going to grow with asynchronous maturation of nucleus to the cytoplasm. So we are seeing that the nuclear maturation will lag behind. It will lag behind as the cytoplasm matures. So that asynchronous maturation is what brings about megaloblasts. This one results into megaloblasts in the bone marrow. So we find the megaloblasts instead of normoblasts in the bone marrow due to impaired nuclear maturation and cell division. Because we know that vitamin B12 and folate, they are the one responsible for biosynthesis of DNA. So without them, the nucleus does not mature. So we are saying it is due to impaired DNA synthesis leading to lagging behind of nuclear maturation compared to the cytoplasmic maturation, resulting into formation of megaloblasts within the bone marrow. And the causes are key. The causes of megaloblastic anemia is majorly due to vitamin B12, which we can call cobalamin, and number 2 is folate. So these are the major causes of megaloblastic anemia, that is vitamin B12 and folate. And under this section, we shall see causes of these deficiencies. So there are two major causes of megaloblastic anemia, that is vitamin B12 deficiency and folate deficiency. But under here we shall see what causes vitamin B12 deficiency and what causes folate deficiency. So before we move on, we are going to look at how is vitamin B12 absorbed after being eaten. We are going to see sources of vitamin B12 is purely from animal products, which can be liver, can be from meat, from dairy products like milk, from eggs. So purely it comes from animal products. So meaning people who don't take any animal products, like pure vegans, vegetarians, they tend to suffer from megaloblastic anemia. So because its source is only from animal products. Whereas the source of folate, for it, this one can be got from vegetables, fruits, can get it from legumes, you can get this one from also animal products, and so forth. So those are the sources of folate or vitamin B12. So when I take food containing vitamin B12, if I take food and this food contains vitamin B12, which we can call cobalamin, this vitamin B12 is taken through the esophagus, and when it reaches in the intestines, this vitamin B12, the parietal cells, the stomach has what we call parietal cells, and these parietal cells in the stomach will release a factor known as intrinsic factor, abbreviated as IF, and this intrinsic factor released from the parietal cells of the stomach will come and bind to vitamin B12. So when they bind together, they form vitamin B12 intrinsic factor complex, and it is this complex, which is the vitamin B12 intrinsic factor complex, moves up to the ileum, and when it reaches in the ileum, it attaches on its receptor, and these receptors, which I have zoomed here, they are known as tuberine receptors, or you can call them R receptors. So these R receptors, this complex of vitamin B12 combined with intrinsic factor, the intrinsic factor comes and attaches. So the intrinsic factor attaches on its R receptors carrying vitamin B12. And after attaching on this receptor, it engulfs, the cell engulfs the vitamin B12 into the endothelial cell. So vitamin B12 is taken into the endothelium or the cellular cells of the ileum, and after reaching within the endothelium, within the cells or the mucosal cells, it is bound to transcobalamin. TC is going to be, TC is an abbreviation of transcobalamin. Transcobalamin. This is a carrier protein. Transcobalamin is a carrier protein. So after intrinsic factor attaching on the R receptors in the ileum, it enables entry of vitamin B12 into the mucosal cells, and when they reach there, they bind with transcobalamin type 2. It is transcobalamin type 2. And this transcobalamin, which is a carrier protein, is the one that carries vitamin B12 into the portal saturation. This is the portal saturation. And when it reaches in the portal saturation, transcobalamin vitamin B12 complex is taken to the liver, and other reticuloendothelial cells like bone marrow to be utilized. So that is what we can discuss under absorption and metabolism. This is absorption of vitamin B12, which we know the most active form of vitamin B12 is methylcobalamin. So this vitamin B12, which we can call methylcobalamin, bound to transcobalamin in the cells, that is in the bone marrow or portal saturation, is its active form. This is the active form, which is methyl. The active form is not this one. The active form is methylcobalamin. So this methylcobalamin, which is the active form, we are saying this is the active form of vitamin B12. It is converted to methyl tetrahydrofolate, and this methyl tetrahydrofolate will remove a methyl group, whereby when we have homocysteine, we are going to add this methyl group on tetrahydrofolate to form methionine. And this reaction occurs in the presence of vitamin B12. So vitamin B12 is acting as a cofactor due to conversion of methyl tetrahydrofolate to tetrahydrofolate, which is a precursor for formation of deoxyuridine monophosphate and deoxythymidine monophosphate. And these ones together are the one that results into DNA synthesis. These ones results into DNA biosynthesis. They are precursors whereby they are responsible for formation of purines and pyrimidines. So when we form purines from these, which are nucleotide bases, we are able to synthesize DNA in the presence of DNA polymerase. So that's why we said in the brief is due to impaired DNA synthesis. So meaning if I do not have vitamin B12, I cannot form tetrahydrofolate, which is a precursor for formation of DNA. So meaning without tetrahydrofolate, we cannot form DNA. And the pathway that is for vitamin B12, then for folate also, when taken, this folate forms what we call folate polyglutamates. And these folate polyglutamates, after being absorbed in the ileum, they are the ones that are converted into tetrahydrofolate. So meaning, still, folate also ends up forming tetrahydrofolate, and this tetrahydrofolate can be converted to dihydrofolate, and it can be also converted to, it can also be going to under the same mechanism, and still it is used for DNA synthesis. For how? By forming purines. This one results into formation of purines. So when we have folate, after being taken, it is used via dihydrofolate forming tetrahydrofolate, then forming purines, which are responsible for formation of nucleotide bases that the DNA polymerase uses to synthesize DNA that is important in erythropoiesis. So after seeing the absorption and the metabolism of vitamin B12 and folate, which we have said even in different sources, that this is animal products, this is both plants and animal products. And we have seen the intrinsic factor. So at the long run, we shall see among the causes shall be deficiency of intrinsic factor. Because without intrinsic factor, we cannot absorb vitamin B12. Because without it, it cannot attach on the tuberine or other receptors. And we are going to see even people who have deficiency of transcobalamin 2, meaning they cannot also transport vitamin B12. So we shall see some of the causes under there. So this one leads to causes whereby cause number one, we are going to categorize them into three. Either one can be due to decreased intake, because of folate or vitamin B12 deficiency can be decreased intake. Number two, it can be malabsorption. Malabsorption is another cause. Or if it is not malabsorption, the third one, it can be increased demand. Increased demand. And even we shall see number four, some drugs. So, under decreased intake, if someone is taking food which is deficient in vitamin B12, for example, typical vegetarians, it will lead to this megaloblastic anemia. And also people who are taking foods without folic acid. So this is a decreased intake. Then under malabsorption, we're going to see people with gastritis, due maybe to chronic gastritis, due to chronic maybe peptic ulcer disease, it can lead to this. We are going to see people who lack intrinsic factor, lack of intrinsic factor, that is pernicious anemia. Pernicious Anemia. Pernicious anemia is caused by lack of intrinsic factor of Castle. And this intrinsic factor of Castle, we saw it is produced by the parietal cells of the stomach. So if someone lacks intrinsic factor, maybe due to autoimmune antibodies that come and destroy the parietal cells, or due to chronic gastritis, this one is what we call pernicious anemia. So it is also a cause of megaloblastic anemia. Another thing we can see is increased bacterial overgrowth. Bacterial overgrowth in the ileum, in the GIT. And this bacterial overgrowth brings about competition for the vitamin B12. So you find you're competing with the bacteria for utilization of vitamin B12. Another thing is parasitic infections. Parasitic infections like fish tapeworm, which we normally call dilatum. Dilatum also consumes, it competes with you for vitamin B12. So we find if we have a fish tapeworm inside here, before this vitamin B12 is absorbed, it consumes it. So little is absorbed. And even other people with bowel syndromes, which can lead to loss of mucosal receptors, any bowel syndrome which can lead to the loss of these receptors for intrinsic factor, can lead to a megaloblastic anemia. Then maybe other causes can be Crohn's disease. Crohn's syndrome can also do that, and so forth. Then under increased demand, we can see pregnancy. Pregnancy, there is increased demand, even in infants. When some people are young, due to increased demand, it can lead to deficiency of these factors. Then drugs like methotrexate, which is an anti-cancer drug which inhibits folic acid synthesis or metabolism, can be one of them. Then we have anticonvulsants like phenytoin and phenobarbital. Phenytoin can do that, and phenobarbital. You can see some drugs like sulfonamides doing this, and even alcohol. For the sake of folic acid, chronic alcoholism and liver disease can also be part of the cause. That is for folic acid deficiency. Alcoholism and liver disease can also do that. So these are the causes of either vitamin B12 deficiency or folate deficiency. After looking at the causes, now we can look at the signs and symptoms. How do you know that someone has megaloblastic anemia? And one of the signs and symptoms of megaloblastic anemia, we know there is the common ones, the general ones, that is tiredness or weakness. We have paleness of the mucous membranes. We have headache. We have palpitations. These are the general signs of anemia. But the peculiar ones we are going to see under this session, these people will have what we call neuropathy. This special vitamin B12 deficiency leads to peripheral neuropathy, and this peripheral neuropathy can result into numbness, that is tingling sensations in extremities, tingling sensations in the limbs, you can feel it. Then it can also result into glossitis, that is atrophy of the tongue lamina, glossitis. So these are the signs and symptoms of megaloblastic anemia. So after looking at that, in children we have seen that's why we give pregnant mothers folate and vitamin B12 to prevent neurotube defect. If people are deficient of this, it can lead to neurotube defects like spina bifida. Spina bifida is also a common cause due to deficiency of folate or vitamin B12, which impairs DNA synthesis. So these are the causes, then the signs. So after looking at that, now lastly we are going to see how can we diagnose or how can we confirm that someone has megaloblastic anemia. Hope you are enjoying, hope I'm not confusing anyone. Let us continue learning, but remember to like the video and subscribe if you haven't. So one of the diagnosis, the diagnosis approach of megaloblastic anemia, we are going to start with complete blood count. We see how it looks like. We go to peripheral thin film, we go to bone marrow, and lastly the biochemicals which we can perform on someone's blood suspected of megaloblastic anemia. And under CBC, we are going to look at hemoglobin concentration, which is always between 5 to 10 grams per deciliter, meaning HB0. Then even hematocrit, or what you can call PCV, is also low. It is decreased. Then when we talk about mean cell volume, mean cell hemoglobin, and mean cell hemoglobin concentration, this one is greater than 100 femtoliters, meaning it is greater. Then this one, MCHC, is greater than 32 picograms, meaning there is some small increase in hemoglobinization. Then MCHC is normal. This is under CBC. Why is MCHC normal? The concentration of hemoglobin, there is just a slight increase in the concentration of hemoglobin. So MCH goes high, but MCHC remains normal. So this is high, this is high, these ones are low. That's why we say they are macrocytic but normochromic, because the MCHC is normal. That is under CBC. So if you see someone with this, and sometimes in advanced stage or in severe stage, in advanced stage of megaloblastic anemia, it can lead to what we call pancytopenia, whereby under pancytopenia, we see low levels of white BC also, and low levels of platelets, plus low levels of RBC count. All of them are low. That is in severe stage of megaloblastic anemia, which is a peculiar thing of pancytopenia, meaning decreased levels of all cell lines. That one is in severe megaloblastic anemia. Then under peripheral film, here we shall find that the cells are macrocytic. We shall find there macrocytes, microcytic, normochromic cells. Macrocytic because MCV is high, but normochromic because there is slight increase and MCHC is low. Then we shall also see some marked anisocytosis. There is marked anisocytosis, that is variation in size due to variation in the size of cells. We find this majority are macro, they are bigger, but some are also small and some are normal. Like if we draw in our peripheral film, we find this is a lymphocyte, small resting lymphocyte. So if I compare with the nucleus, you find that these ones, they are big but oval in shape. So this is what we call macrocyte oval. So these macrocytes, they are oval. This is what distinguishes from non-megaloblastic, where for them they are round in non-megaloblastic, but in megaloblastic we see poikilocytosis of ovarocytes. There is poikilocytosis of ovarocytes. So they are big in size, greater than the nucleus of a small lymphocyte, or greater than 8, but at the same time they are oval in shape. So you find another one is like here, also very big, ovoid in shape with hemoglobinized area, but because of anisocytosis you find others are normal. Find some are normal. And then another peculiar thing we are going to find is if this is a neutrophil, this is a neutrophil, with granules, we shall find the neutrophil is hypersegmented. We shall find hypersegmentation of the neutrophil, whereby the neutrophil has greater than 5 lobes. Greater than 5% have lobes greater than 5. And that is what we call hypersegmentation of neutrophils. So we find there hypersegmented neutrophils, or you can call it right shift of Cook and count. This is right shift of Cook and count. That is, and this is the most common feature in megaloblastic anemia, where we see hypersegmented neutrophils, whereby the neutrophils, the normal ones have 3 to 5 lobes, but you find these ones, the majority of them, they have 1, 2, 3, 4, 5, 6 lobes greater than 5. That is what we call hypersegmented, which is a peculiar feature. So under PBF, we are looking for ovoid macrocytes, is peculiar, together with hypersegmented neutrophils. So these are the key features we are looking for in the PBF of megaloblastic anemia. So after seeing that under bone smear, in the bone marrow smear, we can look for, we shall find that is hypercellularity. The cells are enough. Then when we do Paris Prussian blue, it will be positive, meaning there is increased iron stores are enough. There is enough iron stores. Another thing which we can see under bone marrow smear is we can look at the presence of megaloblasts. Presence of megaloblasts, which are seen, they are different from normal blasts because they have impaired DNA replication or mitosis, so there are many tubular fibers. But there is one thing we have not talked about under PBF. Those are red cell inclusion bodies, whereby under red cell inclusion bodies in the PBF, if I can put it somewhere here, still in the PBF, we shall look at red cell inclusion bodies. Inclusion bodies, especially the inclusion bodies in people with megaloblastic, we can see basophilic stipplings due to ribosomal DNA. Then we can see Howell Jory bodies, and we can also see Cabot rings due to remnant of spindle fibers. So you can find them. Basophilic stipplings can be there. You find red cells with dark blue staining granules, that is basophilic stipplings, or you find another cell with the figure 8 pink staining, figure 8, that is Cabot ring due to remnant of spindle fiber, or you find a cell with a Howell Jory body, dark blue staining, body eccentrically located. This is what we call Howell Jory. So these ones can also be found in megaloblastic anemia under peripheral thin film. Don't forget those inclusion bodies. Then we have seen bone marrow. Then another thing we can talk about is the biochemicals. And under biochemicals, we can determine serum vitamin B12, stroke folate. And if there is megaloblastic anemia, we find they are decreased, whereby serum folate or serum vitamin B12, they are low. That is, for this one, it means it is less than 180 nanogram per liter, whereas this one is less than 150 nanogram per liter. Then we can also do another test known as serum homocysteine. Is also, because we saw here that in the presence of vitamin B12, homocysteine is converted to methionine. So meaning when there is no vitamin B12, there is increased levels of homocysteine. So in megaloblastic level in anemia, we find increased levels of homocysteine. And apart from that, another thing we can talk about is the deoxyuridine suppression test. Deoxyuridine suppression test can also be done. And if it is suppressed, then it means there is deficiency. And another thing we can do is the serum lactate dehydrogenase, LDH, due to increased breakdown of these megaloblasts. We shall see in the bone marrow, due to presence of megaloblasts, these megaloblasts are destroyed within the bone marrow. And when they are destroyed, they can lead to increased levels of LDH. So you find LDH levels, they are high. And another thing we can find under biochemical test, we can also do serum bilirubin. And this serum bilirubin will be moderately high. It is moderately high. Why is it moderately high? It's due to breakage of megaloblasts in the bone marrow. So this increased breakdown of megalo cells results into moderate jaundice, which is characterized by hyperbilirubinemia. Those are also another test you can do. Then if you are suspecting pernicious anemia to be the cause due to lack of intrinsic factor, you can do Schilling's test, whereby you give radiolabeled. Here we give radiolabeled vitamin B12. Vitamin B12, which is radiolabeled. Then you see how it is able to be, if you have given 10 milligrams of vitamin B12, after 2 hours, you come and visualize how many of the radiolabeled vitamin B12 are absorbed into the bloodstream and the saturation. That is what we call Schilling's test. Or you can do even intrinsic factor antibody test to rule out autoimmune disorder of the antibodies that destroy parietal cells or that destroy or inactivate intrinsic factor, which does not enable absorption of vitamin B12. We can also do endoscopy to rule out gastritis, depending on the cause. We can also do stool analysis to rule out fish tapeworm, to look for the dilatum eggs. Also, we can do that. Then bacterial overgrowth and so forth. So these are the lab tests we can do to confirm megaloblastic anemia. Hope you have gained a lot from this session, and we urge you to always continue to support us by subscribing, liking the video, and sharing. Thank you so much, and may God bless you.